this lecture is going to look at cancer. So in the first lecture out of Chapter 8, I talked about telomeres and how telomeres can create immortal cells. And in this lecture, I'm going to talk more about how those cells can then turn into cancer. So cell division out of control leads to cancer. However, not all immortal cells or cells that are undergoing this out of control division necessarily turn into cancer. And so we're going to talk about that. So cancer is going to be unrestrained cell growth and division, but it's very specific. So we'll talk about that. It can lead to tumors, and the tumors have to have specific characteristics in order to be classified as cancer. And it is the second leading cause of death in the United States. The first, the top leading cause is currently heart disease. All right. So these are two, pretty big problems for us. The thing is that for the United States, for it to be cancer and heart disease, those are two diseases actually of older populations. So that's part of the problem, is that cancer is one of those diseases that shows up later in life generally, which is why evolution has not acted upon it, right? If you're wondering about that. So I'm not saying that it's something that we can't work on. There are lots of researchers working on trying to figure out how we can address it. But let's take a look at what creates cancers. And so thing is that cancer cells have several features that make them different from normal cells. So the first one has to do with this. Normal cells, when they're growing and dividing, they will stop dividing once they are touching a certain number of cells on every side. So the way I think about this is cells have personal space that they observe. And so when there's enough of them around, they're like, okay, guys, we're good. Nobody else divide. We call this contact inhibition. And so you can see here they're growing cells in petri dishes. And so normal cells, they're just going to create a nice flat growth of cells. On the other hand, cancer cells, they're going to not have any contact inhibition, meaning that they really don't have a sense of personal space in a way. So they just keep dividing, even though they're just going to pile up and group together. So in a petri dish, they're going to pile up on top of each other. If they're inside of, say, a tissue, they're just going to create these big groups and lumps that can turn into those tumors. So they're going to basically work almost kind of against themselves, because the reason why normal cells do this is because it helps as far as resources. They're not going to compete with each other for resources as much as something like this. Okay. The next one is related to those telomeres. So normal cells are going to eventually no longer be able to divide. They're going to get that signal to stop and self-destruct, because they'll start cutting into the telomeres and they can't rebuild them. On the other hand, cancer cells, they never lose their ability to divide. They can do so infinitely, and as they do so, continue to build up mutations, right? The last one, and this is what is tied to what we consider cancer, and that is that they have reduced stickiness. Now that sounds weird, but normal cells have basically kind of like Velcro almost. So they tend to stick together and stay in one place. However, cancer cells lose this sticky ability. So they break off and start floating around. And this allows them to travel to different areas in the body. Okay, so they can have a tumor, but when they have this reduced stickiness, they then can leave that tumor and move to other areas. So those are some of the features that cancer cells can have. There are other ones. So for example, some of these tumors eventually can get the ability to get the body to grow them blood vessels so they can have their own blood supply. Not all of them have that, but these are generally the characteristics we see with what we consider cancer. So tumors are going to have the first two characteristics. For sure, benign tumors. Benign tumors have not gotten that last characteristic of the reduced stickiness. Benign tumors are not considered cancer. These are ones that can be removed safely because they are located in a central area. And sometimes, like I know there's things like fatty lymphomas and moles and things like that are considered benign tumors, and this is because they haven't moved. They're staying in one spot. On the other hand, malignant tumors, this is where we now consider them cancerous because they've undergone metastasis, meaning that they have lost their stickiness. They have now started to travel to other parts of the body. And so you can see here, each of the white spots in this, sorry, it's not an X-ray and I can't remember what it's called off the top of my head. But each of the bright white spots in this diagram are cancer cells that have spread via circulatory system, lymphatic system, because those cells have broken off and have moved throughout the body. This is much more difficult to treat and becomes much more dangerous. So then the question is, how does cancer kill organisms? And we'll get to that. I do have a question. So, which of these below is false about that's supposed to say a cell, or no, it's supposed to say about cells in a malignant tumor. That A isn't supposed to be there. So which of these is false about cells in a malignant tumor? And here, the best answer is C. Right? Because a malignant tumor, these guys have migrated away. Benign tumors, they stay put. We'll talk about D. I know we haven't talked about that yet, so you may have said that one, and that's okay. They definitely are going to lack contact inhibition because that's what's allowed them to create that tumor. And they lack that cell cycle control because that's part of what has allowed them to divide uncontrollably. Okay, so let's talk about what cancer is. How does it usually cause death, especially if it goes untreated? So here is a tumor. In this case, they're showing it in a lung, right? And what are these cells doing to this space in the lung as they multiply? Hopefully you can see, and you said something along the lines of, they're pushing up against everybody else. If you notice, it's pushing up against these vessels, and it's going to be pushing up against the other cells. And that's exactly the problem, is that these tumors are going to start to disrupt function in the area that they're in. They're going to take nutrients and supplies and energy away from the cells and tissues that they're in because there's so many of them. They need more material generally. And so as that tumor gets larger and larger, it becomes more and more problematic. right? And so eventually this can cause death, especially once it starts to metastasize, because now they're going to move into other areas in the body and do the same thing. Okay. So how do we treat cancer? So what are some treatments that we have for cancer? Yeah. So we have things like radiation and chemotherapy. So radiation, this is where we're going to use literally radiation and focus it at the tumor. The idea is that we're going to basically scramble the DNA of those cells, and the idea is to kill the cells. And then chemotherapy, those are drugs that are going to work to disrupt cell division. Because one of the things that we want to do is slow the growth of the tumors, which, you know, is great, but it slows the growth of the cells in the entire body. And so that's why very often the treatment for cancer is often considered as bad as the disease, because what happens for individuals who are undergoing radiation or chemotherapy? You might have said something along the lines of, you know, things that like they lose their hair. It actually might increase like things like bruising. They have problems eating. They feel tired all the time. The thing is that the treatment can actually cause a lot of problems, because those treatments are stopping or slowing cell division for the tumors, but it's also doing the same thing to the patient for their normal everyday cells. So, all right. So that's it for cancer. The next lectures are going to be on meiosis.